Hi, my name is Peter and I'm the Technical Product Manager for Bactobox at SPT Instruments. This video is our quick start guide and the best way to get familiar with Bactobox. Installation qualification, also known as IQ, is the steps you take for verifying that the instrument has been properly delivered, installed and configured. Here we will only go into the hands-on aspects, but for a full checklist IQ, please contact your local sales representative. First, let's do the unboxing. Bactobox comes in a foam padded cardboard box, which is good for long-term storage and shipping. Here you find the safety instructions. You find the Bactobox with display protector. You find a USB kit for software updates and to retrieve data. You find a tubing kit and finally the power supply with adapter. First, let's mount the tubing kit. The black part is the outlet and mounts directly on the black panel mount on Bactobox by Luer lock connectors. Finger tight is fine, no need for tools here. The white part is the inlet and it comes with a 40 micrometer pre-filter already installed. To avoid tangling, unscrew the pre-filter and mount this to the white panel mount. Subsequently, mount the inlet part to the pre-filter. Next, plug in the power supply. Make sure to press the power plug firmly into place until the plastic is flush with the box. Otherwise, you may experience annoying restarts if you fumble with the power cord. Flip the rocker switch to turn on Bactobox. The boot process takes about a minute. Now it's time to install the flow cell. This is the heart of Bactobox and where all the magic happens. Follow the on-screen video to dock the flow cell into place. First, let the flow cell sit loosely on the mount and then press firmly on the upper part until you hear a distinct click. Then click the lower part into place. When the flow cell is docked correctly, it should give a springy tension when you press gently on the middle part. This Bactobox has been inactive for more than a week and therefore we're prompted to do several things on startup. First reminder is to perform a QC test. This is short for quality control. We'll do this in a minute as a part of the operational qualification. Here, just press stop to dismiss the screen. The stop button generally cancels any running program and is therefore the safest way to dismiss notifications and get back to the home screen. Second reminder is to perform a clean on a disinfection vial. You'll want to perform frequent disinfection of the system to make sure the bacteria you detect are from the sample and not background from the system or the previous sample. To do a clean, transfer the tubing kit to a fresh disinfection vial and press the clean button. The rack can be tilted so it's easier to transfer the tubing kit from tube to tube. It's important to pull by the rubber stopper and not the tubing, as otherwise the connection may become loose. In the first part of the clean program, a peristaltic pump creates negative pressure and transfers the disinfection liquid from the vial through the inlet steel pipe. After a while, the liquid starts to drip from the outlet, and in the end of the clean cycle, the flow is reversed so air is used to empty the flow path. After 1 week of inactivity, it's recommended to run a flush program on a deep clean vial to degrade biological substances in the system. The flush program is found within the menu. Press and hold the measure button for 1 second and navigate down to the flush program. Now it's time to test if the instrument and flow cell are working as they should by performing an operational qualification, also known as OQ. Again, here we're just showing the hands-on parts, but for a full checklist OQ, just contact your local sales rep. The OQ consists of 2 steps. First, measurement on a QC test, and then measurement on a blank sample. First, we do the QC test. This program serves 2 very important purposes. First, it checks if the impedance fingerprints and absolute concentrations are within specifications. And secondly, it adjusts the pump speed by adjusting for optimum flow rate. We use a well-defined suspension of 1 micrometer silica beads to simulate the size of bacteria. We supply 2 types of silica bead formulations. The flask contains sufficient volume for 99 QC tests, while the plug cap vial is pre-pipetted and intended for a single measurement. Both options must be diluted 1:100 prior to use. If you prefer the flask option, it's extremely important to shake it vigorously 100 times to suspend the sedimented silica microspheres. Subsequently, use reverse pipetting technique and a P200 pipette to transfer 101 microliter to 10 ml of ready-made Bactobox diluent and vortex for 30 seconds. Please store the flask upright after opening, as otherwise beads may be lost in the threads over time. If you prefer the single-use option, it's extremely important to centrifuge vials at 4000 Gs for 1 minute before opening the vial. Otherwise, beads might be lost when the cap is removed. It can be slightly challenging to remove the plug cap, so we've included a tool to help you. 101 microliter has already been pipetted by SPT, so you can just do the 1:100 dilution by transferring the contents of a ready-made dilution vial into the vial, followed by 30 seconds of vortexing. Make sure you see a vortex forming. Sometimes it may help to invert the tube when vortexing. The QC test sample is ready, and now we run the QC test. It should always be done on a freshly vortexed vial, as the microspheres will sediment over time. Press and hold the measure button for 1 second. We've already done a clean, so we can skip directly to transferring the tubing kit to the prepared QC vial. Press OK to execute the QC test program. In the starting up period, the flow rate and the conductivity of the sample is checked. Given that these are within the specifications, the actual measurement will start and results will appear after the measurement. Here, the concentration is 2.3 million particles per ml. The certificate of analysis declares a concentration of 2.28 million silica particles per ml, so we're safely within the accepted 15% range, and the QC test is passed. Several other parameters are checked automatically in the QC test program, and in case something is off, error messages will appear on the display. Consult the troubleshooting guide if you get error messages. Better note this down along with the measurement ID, dilution factor and sample information. The second part of the OQ is to check a blank sample, that is, a Bactobox diluent with no added silica beads or bacteria. First, run a clean program to remove residual silica beads. Next, transfer the tubing kit to a vial with Bactobox diluent. We recommend that you use the SPT diluent when running a blank sample, as it's been tested to be sterile and free of particles. For a passed blank test, the intact cell concentration must be below 5,000 intact cells per ml, and the total particles must be below 100,000. The detected particles usually originate from the pump. The rollers in the pump head squeeze the peristaltic tubing to drive the flow. This may lead to shedding of microplastic objects from the tubing. But because the impedance fingerprint for bacteria differs from microplastic, the background is not a problem for bacterial enumeration. Both the QC test and the blank test was within specifications, so we can conclude that the device is operating as it should. Better note this down along with the measurement ID, dilution factor and sample information. In the OQ procedure, we advise you to use our ready-made diluent, but for routine enumeration of bacteria, it is cheap and simple to prepare the diluent yourself. Bactobox uses an electrical detection principle, which means that it is very dependent on the conductivity of the sample. If conductivity is too low, the electrical currents will be weak and we risk underestimation. If conductivity is too high, we can no longer distinguish bacteria with intact membranes from other objects. The accepted conductivity range for the default measurement is 1600 to 2100 microsiemens per centimeter. To have some wiggle room, it's best to aim for a diluent conductivity close to 1900 microsiemens per centimeter. Usually a 1 to 9 dilution of PBS will work, but you may need to tweak this slightly as conductivity depends on temperature and the PBS recipe. Transfer 10 ml of 1x PBS to a measurement cylinder. Then add 80 ml of low conductivity water like milliQ or water for injection. This solution likely has the right conductivity, but it also contains bacteria and other particulates that we need to remove, so we'll only be analyzing the bacteria in the samples of interest and not the background from the diluent. Remove objects by sterile filtering through an 0.2 membrane filter. Here I use a syringe filter, but if you need to prepare larger volumes, it's more convenient with vacuum filtration. I install the syringe filter on the luer lock. Pour the liquid in and reinstall the plunger. Next, I sterile filter in aliquots of 10 ml, so they're ready to use just like the gamma sterilized SBT vials. We recommend that you sterile filter fresh on the day of use, so there is no risk of growth or background in the diluent. Now it's time for the moment of truth, where we check if the conductivity is appropriate. Perform a clean on a disinfection vial. Transfer the tubing kit to a sterile filter diluent tube. Press and hold the measure button and find the conductivity program. Here the conductivity is 1822 microsiemens per centimeter. This will be the conductivity of a blank sample after a disinfection vial clean, where traces of isopropanol is left in the flow path. Sometimes we'll be doing bacterial measurements where isopropanol is not present in the flow path. So let's do a second measurement to check the conductivity when the previous sample was the diluent. Press and hold the measure button and find the conductivity program. Here the conductivity is 1897 microsiemens per centimeter. Both conductivity values are perfectly within the range from 1600 to 2100 microsiemens per centimeter, so we are happy with the diluent. If it was outside specification, we recommend trying with a 1 to 10 ppс. Please consult the protocols booklet for more information. Let's move on to the interesting part with analysis of real bacterial samples. There are 3 prerequisites to keep in mind to get reliable Bactobox results: conductivity, linear measurement range, and single-cell suspension. Even though we thoroughly check the conductivity of the diluent, the conductivity will also depend on the dilution factor for the given sample. I would like to briefly show you the influence of broth on your prepared sample. Here I've done a factor 10 dilution series of the growth medium. When I check the 10 to the minus 5 dilution, it hits a perfect conductivity. Same thing for the minus 4 and minus 3 dilution. But when we go to the minus 2 dilution, we get somewhat higher conductivity. This is because the TSB medium has a conductivity of approximately 16,000 microsiemens per centimeter, and when diluted 1 to 100, it will add additional 160 microsiemens per centimeter to the diluent's own conductivity. This one will likely be too high. If we go to the minus 1 dilution, it is clearly out of specifications and therefore not suitable for Bactobox measurements. Same goes for the growth medium. In summary, conductivity-wise, we should be able to measure all samples above dilution factors of 100. Next prerequisite is the linear measurement range. The quantification range is 10,000 to 5 million total particles. A typical overnight culture will be around 1 billion intact cells per ml. This is way too high, and we need to dilute it prior to performing Bactobox measurements. The dynamic range of Bactobox can handle jumps in step of 1 to 100 dilutions instead of the 1 to 10 dilutions that are traditionally used in microbiology. With a 1 to 100 dilution, the sample will still be too concentrated. But with a 1 to 10,000 dilution, we should be safely within the Bactobox quantification range. The final prerequisite for optimal results is a single-cell suspension. Bacteria tend to stick together and form clumps. If no disaggregation is performed, multiple bacteria will be counted as one or not detected at all because objects bigger than 5 micrometer won't enter the measurement channel. For reliable results, it is extremely important to disaggregate the sample. In most cases, beading or vortexing with 3 mm glass beads is sufficient. Clumping will also be a problem for other enumeration techniques, such as plate counts. Okay, let's see how all these aspects work in practice. First, I disaggregate the sample by vortexing with glass beads for 1 minute at max speed. The bacterium we have here, Staph epi, is usually relatively easy to disaggregate. After the disaggregation, I do the first 1 to 100 dilution by transferring 101 microliter of the disaggregated culture to 10 ml of diluent. Remember to vortex. And finally, I do another 1 to 100 dilution to reach the final 1 to 10,000 dilution. Vortex again. Let's clean the device to make sure no background is present in the system. Next, simply place the sample in the rack, transfer the tubing kit to the 1 to 10,000 dilution, and click Measure. As with the QC test, there will be an initial starting up phase where flow rate and conductivity is checked. If both parameters are within specifications, we proceed to the measurement phase and wait for the results to appear on the screen. Here, the full measurement time was used and the intact cell concentration was 79,000 out of 94,000 total particles per ml. Multiplying with the dilution factor 1 to 10,000, we see that the bacterial concentration in the shake flask is 0.79 billion intact cells per ml. The total particle concentration is in the lower quantification range. The intact cell concentration is fine, but the total particle concentration will be somewhat affected from spallation. Therefore, if we want to get more reliable live dead information and faster results, it's worth trying a more concentrated sample. Let's try with a 1 to 1000 dilution. For this, I dilute the original 1 to 100 dilution tenfold. First, I withdraw and discard 1 ml of diluent. Remember to vortex. And then use the same pipette to transfer 1 ml of 1 to 100 dilution to the remaining 9 ml of diluent. And vortex. Normally, we would do a clean in between different samples to remove carryover bacteria from the previous sample. But because we're going from low to higher concentration, we can skip the cleaning step and proceed straight to measuring the next sample. Notice that the results appeared much faster than with the previous sample. With more bacteria detected, the fast results feature is activated because the concentration is already stable. As expected, the intact cell concentration is about 10 times higher, and at these total particle concentrations, the contribution from non-bacterial objects is practically negligible. Now, the ratio between intact and total objects can be used as a quick and dirty live dead indicator. If you won't be using the device more today, simply run a clean program on a disinfection vial and shut down Bactobox by flipping the rocker switch. Generally, remember to run frequent clean programs. When moving from a sample with more than 100,000 intact cells to an unknown sample, when leaving Bactobox inactive, and prior to shutting down. At this point, I'd like to mention 3 best practices considerations. When screening for the optimal dilution factor, work from low to high bacterial concentrations. This minimizes risks associated with clogging. When you get experience with your specific strains, it will be easier to determine the right dilution. Here, the 1 to 1000 dilution is spot on. Finalize measurements for a given dilution series within 15 minutes. The diluent has less salt than growth medium, which can result in more fragile cells. Working swiftly also minimizes bacterial growth. Never click measure on the same vial twice. If technical replicates are needed, prepare them from the primary sample, that is, the shake flask. This approach includes the variation from pipetting and vortexing. Now you've learned how to measure samples. Next, I'll show you how to extract data from the device. Connect a Windows 10 or 11 PC to the Bactobox micro USB port via the supplied USB cable. The Bactobox will appear as an external drive under this PC. 2 files are seen. Let's have a look on the summary file first. This is an overview of the programs executed on Bactobox. For most Excel versions, the file will open with the data columns immediately. In rare cases, it might be necessary to use the function text to columns. Here we see the different programs that were executed previously. I'm mainly interested in the bacterial concentrations, so I choose Format as Table and apply a filter for measurements only. And sort the measurements so they have the same order of appearance as my lab book. Now I include the data from my lab book for the given measurement ID, description and dilution factor. I also add an additional column to calculate the concentration of intact cells in the shake flask. To get the bacterial concentration in the shake flask, I multiply the dilution factor with the intact cell concentration. Same thing for total particles. Change to scientific formatting. Intact cell concentration is quite similar for the two measurements, but the proportion of non-bacterial particles will usually be lower in more concentrated bacterial samples because the spallation background is reduced. Remember to save as an Excel file, as otherwise the formatting will be lost. The SPT access file leads you to a browser window with many useful features. The most important feature is the software update. Here you can upload software updates with new features from SPT. If you'd like to do deep dives in the generated data in our tool Bactobox Explorer, you can download the individual measurements by specifying the relevant date range. Click once to select the first relevant date and click again to set the end date. Click Download. Also, if you need to share data with SPT or get assistance with troubleshooting, you can use the Send to SPT feature in the Support tab. It's also worth noting that you get direct access to manual, protocols and troubleshooting guide in the Support tab. This concludes our Bactobox Quick Start Guide. If you have any questions, just contact your local sales rep. Happy Bactoboxing!